is Peyton, and I'm a biology major, so I decided to do my project on a coral, Staghorn coral, and the disease that's overtaking it and leading it to extinction right now. So this disease is called white band disease, and the title of my literature review was The Violent Decline of Staghorn Coral Biodiversity, which is white band disease. And the question for this project is, how does white band disease affect staghorn coral reproduction? And the scientific name for this coral is Acropora cervicornis. So these are some pictures of the early stages of staghorn coral necrosis, which is caused by white band disease, which is the bleaching of the coral. So it can start with just some spots over the coral, one spot, or it can start from the bottom and work its way up. And then we start seeing whole sections being bleached, and it's very contagious, quote unquote. It just spreads like rapid fire. So an introduction to the coral and white band disease is that staghorn coral is one of the most biologically diverse ecosystems in the world. It makes these coral banks have thousands and thousands of fish species, bacteria species, algae, plants. It's native to the Indo-Pacific Ocean, but it's suffering critical endangerment because of white band disease. It does have a symbiotic relationship with photosynthetic algae called zooxanthellae, which I will get into later. But that is one of the things that helps this coral reproduce. One of the main causes of the endangerment of this coral is white band disease, which causes the coral to bleach and go into a process of necrosis, which basically just shuts its cells down. And this is due to ocean acidification and microplastic pollution, which we could relate to tourists, since these corals are in touristy areas with a lot of people coming in and a lot of people coming in the water and bringing everything into the water. The coral reproduces asexually, but fish excretion promotes the coral health and growth as it makes it able to reproduce with itself and build its cells. So like I said before, it's home to thousands and thousands of fish species. It brings habitat to so many crustaceans and plants and algae. So the question that I should be asking while looking through all of this research and evaluating it is, are these oceanic destinations suffering from tourism overload? Coral reefs did make minor reparations during our world shutdown from COVID 2020 to 2021. When people were not traveling, these coral reefs started to come back to life, to be a lot healthier. So during this project, I will evaluate the arguments and observations regarding the decrease in coral populations and their failure to reproduce due to the oceanic disease of white band disease. And the information provided will help paint a picture of the importance of the staghorn coral ecosystems and their reproduction and the threat of white band disease that lie in its path. So the oceanic locations of these corals, we see them in the Caribbean Sea and in southern Florida. And all of these locations are very touristy. So that's one thing that you should keep in the back of your mind when thinking about why these corals are suffering so much compared to other corals. So the objective of this research and these methods are to look at the diminishing corals and look at the demonstration of microplastic bleaching and determine the exact rate at which microplastics prevent staghorn coral from reproducing, which we will speak about in methodology, and look at the rate at which microplastics bleach and melt these corals, because this can be applied to marine conservation and other species, and we can look at how to prevent it happening in other species as well, if we can relate it to this coral specifically. So the first topic that we're going to be looking at is the biodiversity of staghorn coral ecosystems. So the picture that I just pulled up is the asexual reproduction of staghorn coral in its process, and these are its little tentacle, and you can see that this branch, that's what they call them, the branch is coming out of this tentacle, and it'll grow to look like one of these mature ones. And you wouldn't be able to see it if you were just looking at it in the water. This is through a microscope. So one of the benefits of these corals and the biodiversity of the coral is the fishery productivity. So we know that these are in the Caribbean and Florida, which are very heavy tourist destinations, but also locals will fish there for income, and they're huge fishery communities. And when the corals are dying, the fish are also dying, and the fish that don't die will migrate to another part of the ocean. So this hurts the locals and it hurts their ability to have a sustainable income. When the oceans are healthy, the land around them is healthy, so if our coral reefs are dying, we have all these other species dying, and it leads to a chain effect, and it makes the land suffer. So when corals fall to extinction, fish, algae, and crustacean species, the ones that rely on the coral at least, will fall directly after. If not, they will move. And this coral bleaching or necrosis from white band disease will lead to species-wide extinction in the next decade. So, next topic is white band disease. So this is just a before and after of a completely disease-ridden coral bank from white band disease. So as you can see, full of color, full of life, and this is basically the skeleton of a coral in its radiolite structure. So white band disease was discovered in the 1970s, and massive amounts of coral have died since then, and the remaining populations have struggled to recover greatly. It's not something that many populations of coral can recover from, as we've seen in the past. So how can genetic diversity protect Staghorn coral from white band disease? And this is a question of natural selection. So if there is a species that is not genetically diverse at all, aka produces asexually like this coral, we do not have much of the gene pool to pull out from. So there is not much resistance in these corals, especially when they're only reproducing asexually. But we can speak about ways that corals can reproduce in coral farms later on that may help this process of natural selection going hand in hand with white band disease. Some of the long-standing corals may be protected from the disease due to their gene flow, which also goes hand in hand with natural selection and this genetic diversity. This coral does not have a lot of gene flow, but some of the coral farms may give it some. So diversification of these corals began over 2 million years ago, but this specific kind of coral, Staghorn coral, is not diversified at all. So how can these scientists promote biodiversity on these coral farms? So again, the topic of genetic diversity. Some banks of coral have been observed closely because of their genetic diversity, and they have shown resistance to this disease, because genetically diverse coral banks have maintained their health despite suffering the same environmental factors such as coral bleaching and rising water and temperatures. And the gene flow of this also alters natural selection. And diverse gene flow with asexual reproduction is extremely difficult. So these are pictures of some coral farms, which, as we see, two branches interconnected with each other. And if it is two different kinds of coral, then maybe an allele or a gene can be reproduced that can withstand necrosis and white band disease. So that is the point of farming these corals, not only to reproduce, but to give strength to them. So, like I said earlier, we're going to come back to this bacterial algae, zooanthellae, which has a symbiotic relationship with the coral, symbiotic and mutualistic in biological terms, and this bacteria provides nutrients for the coral through photosynthesis, and the coral provides habitat and protection for the bacteria. So when the coral is under thermal stress, like ocean acidification causes the water to be a lot warmer, same as global warming, the zooanthellae abandon the coral and they leave it bleached. But coral will die if their thermal conditions do not return to normal, and when they do return to normal, the bacteria will begin to grow on it again and provide health for the coral. So a methodological approach to farming these corals or tracking their reproduction with this disease would be taking two subjects. It would be two Staghorn coral farms or banks. One will be planted in disturbed waters with microplastics present and fish species present, such as an aquarium with microplastics, and the other would be kept undisturbed in a marine aquarium with only the fish species. So one has microplastics and one does not. Researchers would manage asexual reproduction by looking at skeletal growth in X-rays, similar to other coral reproductive studies. They should also measure the coral branches to look at growth in length and not just width, because this also is a telltale sign of a healthy coral. The researcher would measure the new coral branches by millimeters and check for new growth every month for a year. These growth measurements would help scientists track the coral farm and see which is more reproductive in the presence of disturbance and non-disturbance, but both still benefiting from the fish species. So this is just a simple experiment to figure out exactly how much microplastics deter coral from reproducing. And this is just a chart that demonstrates the steps 1 through 5 simplistically. So experimental design on this project, the scientists would collect small fragments of healthy and diseased staghorn coral, place them in the separate tanks with filtered salt water with the same temperature that they were collected in, raise the water temperature in half of the banks to encourage the zooanthellae to abandon it, like we talked about earlier. We would create small lesions on all healthy and bleached coral fragments to increase the likelihood of contracting white band disease, introduce diseased fragments to tanks with healthy and bleached fragments, observe the rate and percentage of tissue loss, and the objective of this project would be to determine the influence of zooanthellae loss on rate of white band disease, spread, amount of tissue loss. So we have a chart here that would look at how tissue loss happens with white band disease with and without zooanthellae bacteria, which promotes healthy corals. So the significance of this research to future projects would be, it would give us more information on the small amounts of coral that have become resistant to white band disease with the help of its mutualistic partner zooanthellae bacteria. There's very little research on corals that are resistant to oceanic disease. It would also help with the recovery of diminishing populations, which is a very, very focused topic right now. And it would help with efforts to diversify coral banks and species of corals with coral farms or without, modification of reef patterns, which is hand in hand with coral reef farming, and then the influence on research of gene flow in asexual populations. It would also help with perseverance and natural selection, which goes hand in hand with the diminishing populations. So, research and literature challenges that I faced during this project are vast. I had a lot of issues with my research and even more with formatting, just because I'm not familiar with MLA, but I will show you my letter of reflection. So I wrote, beginning this assignment, I knew I wanted to take a scientific approach in regard to my major, which is biology, and I already told you guys this. The first challenge that I ran into with choosing a topic was the jargon used in scientific journal articles. I had to either analyze and deconstruct the articles in order to understand them, or choose a topic I had at least a small amount of prior knowledge on, and I combined those two together and I did both. I had a small amount of knowledge on coral reproduction, but I also had to analyze a lot of the jargon used, especially in charts and graphs. Choosing coral reproduction was a strength for my topic of interest, but although I have prior knowledge on the topic, I ran into challenges. One of the topics I had to do extensive research on was the concept of genetic diversity from asexual reproduction. In biology courses, students learn that one of the leading causes of extinction in species, mostly bacterial species, is having little to no genetic variation. Having no genetic variation will cause a species to be too susceptible to environmental threats with no way to expand reproduction. Reading the articles about coral farming and understanding the topic more led me to learn about genetically modified organisms and how using this approach can help save a species. The final issue I had in writing my literature review was formatting, which was an issue I encountered many times throughout the course, simply because I'm not familiar with MLA formatting. And here's my list of sources. They are all scientific journal articles. And thank you for watching.